All right, welcome back. In this video now, we're going to talk about a particular disease you're going to see in your exam. This one is like 90% sure. I get my point. Orotic aciduria. Orotic aciduria, meaning excess of orotic acid in the urine. Excess of what? Orotic acid in the urine. That's what we call orotic aciduria. Orotic aciduria. All right, there's a particular place in the biosynthesis of pyrimidine in which you convert your orotate to OMP, then OMP to what? To UMP. You remember that part whereby, before orotate, it was diiodorotate. Then we got orotate when we now added the double bond there. Then subsequently, we now use our PROPP and convert orotate to orotidine monophosphate, which is called OMP. Then also OMP, using this PROPP stuff. The enzyme we say is what? Orotate phosphoriboside transferase. Good. Now, once we got our OMP, this our OMP can also be what? Be decarboxylated. I say we can lose this CO2, this group here, as what? As carbon dioxide. Is that not so? Can lose that carbon dioxide. When we lost that carbon dioxide, we now got what? UMP. This is a structure of UMP. I told you guys that time that this place here. You are seeing it now. UMP, uridine monophosphate. You can see it there. All right, so that is it. Then this is just the pathway. For orotic aciduria, the enzyme that is defective is this enzyme and this enzyme, these two enzymes. So these two enzymes are also called bifunctional enzyme. There are two enzymes in one. Bifunctional enzyme. Bifunctional enzyme. So orotic aciduria is as a result of a defect in the gene responsible for the synthesis of waiting. This is bifunctional enzyme. The bifunctional enzyme consists of two enzymes. What are they? Orotate phosphoriboside transferase and OMP decarboxylase. Orotate phosphoriboside transferase and OMP decarboxylase. If these two enzymes are defective, the person will come down with what? Orotic aciduria. All right. Now you can see that I said orotic aciduria. What does it mean? It means what? Excretion of waiting. Of what? Of what? Can you see this? Orotic acid in the what? In the urine. So when you have orotic acid in the urine, we call it what? Orotic aciduria. All right, what are the key features? Please look at these key features very well. You can notice that I put as my number one megaloblastic anemia. Megaloblastic anemia. Megaloblastic anemia. Mind you, I did not just say anemia. It is characteristic. Once you see this megaloblastic anemia, you are most likely looking at, okay, the other thing that causes it, but orotic aciduria, the kind of anemia in orotic aciduria is megaloblastic what? Anemia. And this megaloblastic anemia, the normal way we usually treat megaloblastic anemia is by giving vitamin B12 or vitamin B9, folic acid. But in this case, if you are giving vitamin B12 and B9, the patient will not be recovering. So we say it is refractory, meaning it doesn't respond to treatment with vitamin B12 and vitamin B9. I get my point. Okay, that is one of the characteristic signs. Once you as a doctor, you suspect, you run a lab test, then you find out that this person is having megaloblastic anemia, meaning the RBC is having this characteristic sign that is to say the megaloblasts are big. I get my point. You're having all these RBC precursors with large size. So there's a large cytoplasm with a reduced nucleus. So the cytoplasm is bigger because the RNA synthesis is occurring perfectly well. However, the nucleus is being shrinking. So it's not the cytoplasm ratio to the nuclear ratio is too wide. We're having a larger cytoplasm compared to the nucleus. The reason is because the DNA synthesis in the nucleus is not occurring well. Are you understanding my point? So that's why we are seeing that while the cytoplasm is getting bigger, the DNA is not getting bigger, it's becoming even more smaller. So that is what we call megaloblastic anemia. And this megaloblastic anemia, for the case of orotic aciduria, the one you see in orotic aciduria, it does not respond to B12 treatment or B9 treatment. Okay. Another thing again you see in orotic aciduria is growth retardation and failure to thrive. Okay. The main thing I just want you guys to know is that for megaloblastic anemia, it is characteristic of for orotic aciduria, it is characteristic that you see megaloblastic anemia that is refractory, doesn't respond to B12 or folic acid treatment. Okay. All right. What is the cause of orotic aciduria? It's due to a deficiency of this bifunctional enzyme, which we call wetin? Orotate phosphoriboside transferase and OMP decarboxylase. Please note this. So note this. Very, very, very important. Now, okay, you can see that I've already explained it here. I said the megaloblastic anemia is due to wetin? Decrease in wetin? Nucleic acid synthesis, which will now lead to decreased erythrocyte formation. So the erythrocyte, which is the RBC, will be having a bigger size cytoplasm with a smaller nucleus. All right. How do you diagnose it? First of all, you ask for a complete blood count. To detect the type of anemia. Once you see that, oh, it is megaloblastic anemia, you will now check for plasma. You can check for plasma ammonia level, because another thing can also cause this. There's a secondary cause of this megaloblastic anemia, of this orotic aciduria. Not only lack of this bifunctional enzyme causes orotic aciduria. Please note what I'm saying. No. Not only this waiting bifunctional enzyme, not only the lack of this bifunctional enzyme will cause orotic aciduria. Another thing can also cause it. For example, there's one of the urea cycle enzyme, which we call the ornithine transcarbamylase. I pray I'm spelling it well. But ornithine transcarbamylase is a urea cycle enzyme. It can also cause orotic aciduria. I get my point. So for you to confirm if it's actually due to this bifunctional enzyme, you will check for the ammonia level of the plasma. If it is high, then you know that, oh, this orotic aciduria is coming from the urea cycle defect. I get my point. But the one we're discussing today is the one from pyrimidine biosynthesis. Like, this enzyme are defective. However, there's also another secondary cause. This one is called secondary cause. I get my point. But the primary cause is due to this lack of this bifunctional enzyme. The secondary cause is due to the lack of ornithine transcarbamylase. Okay? All right. Now, let's see if there's any other thing here. Okay, notice here. What do we have here? Treatment. How do you treat them? You use you supplement them with waiting. This is the main thing you have to write to. Please, in case they ask you the treatment, you give the patient waiting uridine. Uridine is a base, right? It's a nitrogenous base. So you give the patient waiting uridine as a supplement. So you supplement the patient with waiting with uridine. What will the uridine do? The uridine will be converted to UMP. Have you get my point? Then from there, sorry, uridine is not a base. Uridine is a nucleoside. Let's get that clear. Uridine is a nucleoside. So once I can add phosphate to uridine, what will I get? I'm going to get waiting UMP. If I get UMP, UMP will form waiting UTP. Then also, like, it will first form UDP, then later on form UTP. Once it form UTP, UTP can inhibit waiting carbamoy phosphate synthesis too. Do you remember during the regulation that time? You remember during the regulation of the biosynthesis pathway, we saw that this UTP has the ability to inhibit that enzyme. Let me see if I have that part again. I don't know if I have the part. Okay, let's look at it here. We saw that UTP can inhibit this enzyme. I get my point. So if this enzyme is inhibited, it means that you cannot synthesize the orotate again. And if you cannot synthesize the orotate, then you cannot have orotic aciduria. You won't see orotic aciduria again. So one of the ways in which we treat them is by administering uridine. When you administer uridine will convert to waiting UMP. UMP will form UTP. UTP will inhibit carbamoy phosphate synthesis 2 enzyme. Then from there, we are not going to see any more formation of orotate. So there will be no formation of orotic aciduria again. Then again, this your UTP or this your uridine that you synthesize can also be used to synthesize waiting. Can also be used for to synthesize thiamine, and can be used as a nucleotide also for DNA synthesis. I get my point. Can be used as a nucleotide for DNA synthesis. So this is all about orotic aciduria. Let's just recap it. What do I say orotic aciduria is? Orotic aciduria is a metabolic disorder characterized by high levels of orotic acid in the urine. So you're having orotic acid in the urine. Orotic aciduria, excess orotic acid in the urine. What causes it? Usually, the primary cause is due to the lack of this bifunctional enzyme. What is the bifunctional enzyme? Orotate phosphoriboside transferase and OMP decarboxylase. Good. All right. I said there's also a secondary cause due to waiting ornithine transcarbamylase. ornithine transcarbamylase. All right. Now, what are the key features? The first one is megaloblastic anemia, then growth retardation, failure to thrive. I say the megaloblastic anemia, it is refractory to vitamin B12 and folate treatment. So even if you give your patient vitamin B12 and folate, they are not going to respond. The treatment will not be effective. It won't be working. You will still be seeing the anemia, the reduction in red blood cell size, like reduction in numbers of red blood cells. I get my point. So you're going to see it there also. Okay, what causes the megaloblastic anemia is due to the fact that since we cannot synthesize those pyrimidine nucleotide, I get my point. This enzyme are defective. We can't synthesize the pyrimidine nucleotide, so the nucleic acid synthesis will be what? Will be inhibited. Then the nucleus of the red blood cell will begin to shrink while the cytoplasm is getting bigger. So that is what you are seeing. That is the characteristic sign of the megaloblastic anemia. How do you diagnose it? I said you ask for a CBC count, complete blood count, to detect the type of anemia. When you do that test, you see megaloblastic anemia. Try treating it with this and this, with folic acid and vitamin B12. If it's not working, then you think of orotic aciduria. All right. Then also check for the plasma level of ammonia to differentiate it from waiting from the urea cycle defect, which is this ornithine transcarbamylase defect. I get my point. Ornithine transcarbamylase also cause orotic aciduria. You also see orotic acid in the urine. I get my point. All right. So that is it. That is it. That is for that. I said the treatment, you give them waiting uridine. You give them waiting uridine, okay? You then uridine. Uridine will form UTP, which will inhibit carbamyl phosphate synthesis too. Also, this uridine can be used for nucleic acid synthesis. All right, good. Now, for that secondary cause, that ornithine transcarbamylase deficiency, if you check the urea cycle pathway, like let me spell transcarbamylase deficiency, if this enzyme is defective, that is in the urea cycle only, it will lead to what? We are going to see that phosphate carbamyl phosphate will accumulate, because the function of ornithine transcarbamylase is to convert carbamyl phosphate to ornithine, or I think either ornithine or citrulline. One of those products will be formed. But the main thing is that, okay, so converted to citrulline. Yeah, I remember now. Went to citrulline. The main thing I'm trying to say is that carbamyl phosphate will begin to accumulate. And you know, if you have carbamyl phosphate, you will not be doing more of waiting. You will do more of what? More of pyrimidine biosynthesis. You can go to pyrimidine biosynthesis. And if you undergo pyrimidine biosynthesis, you are going to have more of waiting, orotic acid. So if you have orotic acid, more of it, then orotic acid will not be seen in the urine. So that's why patients with urea cycle defect, like this one is type 2. There are 5 urea cycle defect diseases. This one is the type 2. If they lack this enzyme, carbamyl phosphate will accumulate, which will then be used for pyrimidine biosynthesis, which will definitely form orotic acid, which will not be seen in the urine. These are carbamyl phosphate. Ornithine transcarbamylase deficiency will lead to orotic aciduria also. This one is the secondary cause. Secondary cause. All right. In the next video, we will look at one more disease and we'll call it a day.